Now it's time for the second presentation. And the second presentation is from Vanessa Moll. Vanessa Moll is a PhD student, second year PhD student in the field of analytical chemistry at the University of Innsbruck, Austria. She is part of the working group of Professor Christian Hock and mainly works in the field of near-infrared spectroscopy in cytoanalytics. Vanessa has already worked during her master's study and analyzed aqueous carbohydrate solutions in low millimolar concentration levels. So today, she will tell us about a study of water interactions with solid polymer matrices, which delivers new insight on this topic. And the title of Vanessa's talk is Investigation of Water Interaction with Polymer Matrices by Near-Infrared Spectroscopy. Lawrence, would you please be able to start the video?
Hello everyone, my name is Vanessa Moll and I am from the Institute of Analytical Chemistry and Radiochemistry of the University of Innsbruck. Innsbruck is a small, beautiful city in the Austrian Alps. We recently published our work with the title Investigation of Water Interaction with Polymer Matrices by Near-Infrared Spectroscopy in the journal Molecules, and therefore want to thank our sponsor for the funding. First of all, a little overview about the topics of this presentation. In the beginning, I will give you a short insight about the aims of the study and why we chose this topic. Then I will shortly talk about the methods we used to realize our aims and the experimental setup. Afterwards, I will summarize the most important results and the spectral analysis methods we used in our study. Finally, I will give you a short conclusion and outlook. So, why did we choose this topic? Well, there are plenty reasons. Among others, the interaction of biopolymers with water plays a key role in biological processes. The effect of moisture on commercially used polymers is also of high interest for material science and industrial applications. For example, excess of water may cause swelling and therefore change the mechanical and chemical properties of the polymers. For these reasons, the interaction of water with both synthetic polymers as well as biopolymers is of high interest for a variety of research fields, as well as for industrial applications. The vibrational spectroscopic techniques with infrared and Raman spectroscopy were already often used to investigate the effects of the interactions between water and polymers. In contrast, near-infrared spectroscopy has not yet attracted similar attention in the studies of this problem. Although NIR bands manifest unique sensitivity towards the molecular neighborhood and hydrogen bonding, because NIR vibrations are intrinsically related to the properties of hydrogen bonding existing in the investigator system. Consequently, NIR spectroscopy provides insight into the interaction behavior of polymers and water. The physical principles of NIR spectroscopy make it also very well suited for the analysis of aqueous systems in a practical sense. However, the very high intensity of water bands in NIR spectra can still mask the signal of other compounds present in the sample. In certain applications, this remains to be an unwanted effect, and effective solutions would be helpful. Even though the removal of water bands from vibrational spectra has been studied for years, there is still very little knowledge of universal application in this area. We investigated 9 polymers of varying hydrophilicity, including commercially used polymers, as well as the 3 most abundant biopolymers. In this figure, we ordered the used polymers regarding their approximate hydrophilicity, beginning with the as known most hydrophobic polytetrafluoroethylene to the hydrophobic polypropylene to the slightly hydrophobic polystyrene and polyvinylchloride to the slightly hydrophilic polyoxymethylene to the hydrophilic polyamide 6 and to the hydrophilic biopolymers lignin, chitin, and the most hydrophilic polymer in this study, cellulose, but still non-water soluble. The poor polymers and polymer water mixtures in the concentration range of 1 to 10% of water were investigated by diffuse reflectance NIR measurement with a Fourier transformation NIR benchtop spectrometer. Let's move to the results. The spectra analysis was performed with several methods. Firstly, we performed cluster analysis, where we used the principal component analysis and the median linkage clustering. We also performed regression analysis in the form of the partial least squares regression. As deconvolution techniques, we utilized the multivariate curve resolution alternating least squares and the two-dimensional correlation spectroscopy. We also tested difference spectroscopy in the form of water and polymer difference spectra. Furthermore, we included aquaphotomics and investigated the aquagrams of the polymer water mixtures. On the right side, the resulting NIR absorbance spectrum of polypropylene in the concentration range of 0 to 10% of water is displayed. Here you can see a very nice increase in the water content for the combination and the overtone water band. Please note I will call them like this because this is convention, but they are actually both combination bands. Cluster analysis was performed in order to get a first observation of general trends in between the polymers. As you can see here, the PCA score of the pure polymers reveal perfectly separate groups for the investigated polymers and therefore show that there are significant differences between each individual polymer. Cellulose and chitin here in dark and light blue are aligned relatively close to each other because of their high structural similarity, but still the polymers are easily differentiable. The regression analysis was mainly performed in order to confirm the validity of our experimental setup. Here you can see the PLSR scores from polypropylene in the concentration range of 0 to 10% of water. Also, here the concentration levels are nicely separated from each other. We applied a different spectroscopy approach in order to reveal the NIR line shapes of the water component present in the samples. Water difference spectroscopy is performed by subtracting the spectra of the pure polymers from the spectra of the polymer water mixtures and therefore leaves the water spectra. Here you can see the water difference spectra of polypropylene. It is immediately noticeable that the revealed water bands are very different from that of bulk water here in dark blue. The multivariate curve resolution alternating least squares method was used to separate the polymer water mixture spectra into its spectrum components, the water and the polymer spectra. Here the MCR-ALS spectra of polypropylene water mixture are displayed. The decomposed polypropylene spectrum is almost identical to the original polypropylene spectrum. The decomposed water spectrum in light blue, however, is significantly different from that of bulk water, here again in dark blue. The findings by difference spectroscopy and MCR-ALS method revealed that even very hydrophobic polymers like PP are interacting with water, and that these interactions are clearly manifested in the water component of the NIR spectra. Moreover, a different behavior of both major water bands in the presence of diverse polymers could be revealed. Some polymers, for example, show simultaneous a blue shift for one water band and a red shift for the other water band. Additionally, wave number shifts are much more pronounced for the overtone water band. The individual shapes of the resolved NIR water bands are very different for the investigated polymers. We found polymers of varying hydrophilicity to reveal three major trends. Firstly, hydrophobic polymers like PTFE and PP are non-attracting towards water, and therefore bulk-like water domains are formed more easily in the sample. Secondly, polymers of low to medium hydrophilicity weakly interact with water, and additionally bulk-like water domains are formed. This is the case for PVC and POM. And the third major group is formed by the hydrophilic polymers PA6, cellulose, chitin, and lignin, which are strongly interacting with water, and therefore no bulk water domains are present in the NIR spectra. The two-dimensional correlation spectroscopic analysis is known to be superior in deconvolution of spectra and revealing the effects of intermolecular interactions. On the right side, you can see the synchronous and the asynchronous 2D correlation spectra of polypropylene. By 2D COS method, it was revealed that the sequence of intensity changes of the water bands is different for polymers of varying hydrophilicity. In the presence of hydrophobic polymers, both water bands show the same behavior, while for slightly hydrophobic polymers, the overtone water band reacts more rapidly to the increase in water content than the combination water band. This trend is opposite for hydrophilic polymers. The 2D COS investigations also confirmed the phenomenon that strong hydrogen bonding leads to a weakened band intensity of the interacting species in NIR spectra, which was the case for the biopolymers cellulose, chitin, and lignin. Because the major changes seem to be present in the NIR water bands, we also decided to include aquagrams into our study. An aquagram is a unique way for rescaling the spectra intensity at selected key wave numbers and magnifying small differences. It is therefore useful for estimating intensity trends of large datasets at a glance. If you are already familiar with aquagrams, you may have noticed that we displayed more wave numbers than usual. Aquagrams generally show water patterns exclusively for the overtone water band, but this was not comprehensive enough to describe the differences of the investigated polymer water systems. Therefore, we decided to show the normalized spectra of each polymer in both water regions and did not only display the water or the overtone water band, but also the second major water band, the combination band. In general, aquagrams remain in agreement with the information revealed by the other methods used in the study. But they also revealed unique insights. For example, the existence of water band shifts can be easily monitored in the aquagrams. Moreover, systems which strongly interact with water can be immediately identified by the aquagram. In the case of hydrophobic to slightly hydrophilic polymers like PP, the water component is highly dominant in the aquagram, as you can see here and here. Mostly the significant intensity increase of the water band with rising water content is reflected here. Interestingly, in the case of hydrophilic polymers, like here for cellulose, the aquagram becomes very complex, as you can see here, reflecting a convoluted spectral pattern related to the changing water concentration in the system. The characteristic water bands are not as similar to bulk water in the aquagrams as they are for more hydrophobic polymers. Let's come to the conclusions. The results obtained with the independent methods provide confirmatory results, with each method also contributing unique findings. In this study, we were able to unveil trends related to the chemical nature of the polymer and its increasing hydrophilicity, which are manifested in the NIR spectra. Moreover, we found three major dissimilar behaviors of polymers of different hydrophilicity, which separates the polymers into hydrophobic, slightly hydrophobic to slightly hydrophilic, and hydrophilic polymers. Of particular interest is also the different behavior of both major water bands, the overtone and the combination water band, in presence of diverse polymers. Some polymers even showed a simultaneous blue and red shift for both water bands. Wave number shifts are also much more pronounced for the overtone water band than they are for the combination band. Moreover, the sequence of intensity changes of the water bands is dissimilar for polymers of varying hydrophilicity, while for polymers of weak hydrophilicity, the overtone water band reacts more rapidly to the increase in water content than the combination band. This trend is opposite for hydrophilic polymers. Hydrophilic polymers show in general a more complex interaction with water. Since even the highly hydrophilic polymers like polytetrafluoroethylene interact with water, and these interactions manifest themselves in the water component of the NIR spectrum, the hydrophilicity is therefore not exhaustive enough to describe the interaction of a polymer with water. Taking into account the chemical specificity of the matrix in describing the spectral effects of the water substance interaction is necessary for successful removing of the water contributions in NIR spectra. The analysis of the polymer water mixtures also confirmed that the sensitivity of water towards its chemical environment is a major factor clearly manifested in the NIR spectra. Moreover, with increasing hydrophilicity of the polymer, the spectral manifestation of the polymer-water interaction gets also more complex. To sum this up, we gained new insights into the interaction of water with some specific polymers, but this was just a small piece of the cake, and in order to gain better understanding of the interaction of water with polymers in general, more exhaustive and time-consuming studies will be needed, and therefore, of course, also additional funding will be needed to further investigate this topic. Thank you for your attention. I'm now at the end of my talk, If you want to read the full paper, please scan this QR code. And if you have any questions, please don't hesitate to contact either my supervisor, Professor Dr. Christian Hook, or me by email.
Thank you very much, Vanessa. This was really a fascinating, very, I would say, mind-boggling talk. So the floor is open for questions, but unfortunately, Vanessa and her teammates are not available to answer the questions. But since we have got few minutes for the discussion, so I would like, Yelena, if you would like to answer the question.
Well, thank you, Harper. I would love to. I would love to just at least comment. I'm really sorry that Vanessa could not attend, and I told her that I expect very lively discussion and many questions. And I'm really sorry that at this time they're not here, because we have people in the audience which are doing the polymer water interaction research. And I want to say this was not the only paper published on this topic in this molecular special issue. There was one more paper from Professor Hayashi, but this work was the first that featured aquagrams, actually, showing this structure of water depending in the polymer, in hydrated polymers. And I want to just let all the people know that all these lectures will be available for everyone, and we will make sure that the people get these questions, and we will publish everything on our YouTube channel, so they will be able to comment afterwards on the lecture. So it will all be on YouTube, our academics YouTube channel. And what I wanted actually to comment, let's say from scientific point of view, I just wanted to say that I'm so happy to see this that the sensitivity of this first overtone and the combination band is different depending on the nature of the material, and that is something because that is something that we recently we're showing that these two bands are giving qualitatively different information, and especially this combination band is giving more information on what we usually call moisture or water vapor. And so in this case, this fits very nicely with what we know about materials. And I'm very happy that I could see it here. I'm sorry, that is a short comment from me. I'm certain other people would like to comment. So please, Professor Di Salvo or Professor Tsenkova, if you want to comment more.
Yeah, first of all, hello to everybody. I'm very happy because we have this topic in the webinar. It is so important to know more about protein-water interaction. And it is a very interesting finding, as Helena mentioned. We know it. We realize that the first overtone is different compared to the main band in infrared. Then the combination band gives different information also. And it's normal. It's normal. We will be able to explain details about this. We know, for example, bacteria systems are more active and we can obtain more information in this combination band. But regarding the protein, I think, as Vanessa mentioned at the end, that it is just a start. And I really, really hope that they will continue, and not only the group of Christian Hook, but many other people working with protein will explore
polymers. Sorry,
sorry about that. Polymers will further explore this, because I could see that, for example, we cannot anymore talk about bulk water. I think it's too vague. We have exactly water bands of small water clusters, and they're not clusters. We know that it's a network, but of conformations that we can. They have names, they have bands, they're WOMACs. Now they have WOMACs number, and we can use them to explain lots of new phenomena that we see with polymers, and especially as they started and they mentioned to understand what hydrophilicity means and what hydrophobicity means, how we can explain this from water point of view, not the polymer, but the water point of view, interaction, of course. So what are those specific water species that interact with polymer and define the hydrophilicity or hydrophobicity? I think it's a beautiful venue that aquaphotomics can further expand and explain lots of new things that we really deserve and want to know. Yeah, thank you.
Thank you very much, Professor Senkova. Any other comment? If not, then we're going to move to our next.
I think just a moment. There was Yellen, I think, responded to this low concentration of polymers. Yes. It is possible, and I think it will be even more clear the interaction between water and polymers when they are at very low concentrations, and it deserves studies and further exploration.
Thank you, Professor Senkova.